So we're going to be looking at microevolution today, changes in allele frequencies within a population, and then we're going to be able to use this information to apply it to that Hardy-Weinberg principle. So if y'all remember, evolution is going to be our change in inherited characteristics of a population over generations. So this has to occur over time. Okay, remember, this is not an instantaneous process. Microevolution is the change in our allele frequencies. Remember, frequency is how often is something showing up. Remember, our alleles are our variations. So these are different forms of the gene, blonde hair, brown hair, black hair, red hair, the variations of the gene. And so the frequency, again, is how often that allele is showing up within the population. The gene for hair color is always there. It's how many times you see the percentage that you see the different variations. Okay? And so these things are going to be the results of 4 processes or causes. Okay? So mutation can cause microevolution. Okay? Selection, when there is some kind of preference, whether it's mating, natural selection, environmental selection, gene flow, as well as what's called genetic drift. And we're going to be talking about all these things today, how these things are going to impact these allele frequencies. One of the other things we need to keep in mind as we're going through evolution is that it occurs in the gene pool of this species, not the individual. Okay? The individual does not evolve. Okay? They do not spontaneously mutate and become better suited to their environment. Okay? The evolution is going to occur within the gene pool of the entire species. So let's talk about a small population versus a large population. So with a small population, we're going to see a much bigger impact when something changes. So when these allele frequencies change, their impact is going to be significantly larger in a small population than it would be in a larger population. Also, with a small population, it's easier for them to actually lose genes. If there were only a few copies of the allele in the gene pool, and those particular organisms did not reproduce, then it is easier for the gene to disappear completely from the population. So when a population becomes very, very small, those allele frequency changes have a much bigger effect. It's easier to lose the gene. And so we can see, if we lose these genes, then we can have a loss of diversity. And when we lose diversity, we lose stability, just like we did in ecology. Losing that diversity loses stability. Once an allele has said to become fixed in a gene pool, okay, that means that there is no more variation within that allele. So if the allele is considered fixed, there is no genetic diversity left with that particular allele anymore. There's only one allele left for that particular gene. So let's say we're talking about hair color. If hair color in humans became fixed, that would mean that there's still a hair color gene, it's just only brown. There are no alleles for blonde, there are no alleles for red, there are no alleles for black. There's no genetic diversity there. Okay, so genetic drift was one of the things that we said could cause evolution. So genetic drift is when we have a change in the frequency of the alleles in the population, okay, because that's what microevolution is. So there's a change in the frequency of the alleles in the population due to a random sampling in the absence of a selection pressure. So there's no selection pressures here. There's no preference. There's nobody's better than anybody. And we've got random sampling. Okay, so you can see this happening here in this population of plants. So again, remember, it's a small population. Allele frequency changes have a much bigger impact on these small populations. So if I had generation 1 that has white and red plants in it, and I take a random sample, okay, and those random samples reproduce, okay, I still have a little bit of white and red, but again, I take another random sample here, okay, and so now I'm down to only the red allele here. Okay, so I didn't prefer one over the other. It was just random sampling happening here. And so this genetic drift has an impact when a population is very small. When the population has been dramatically reduced, okay, you'll see the impacts of this genetic drift a lot more. Okay, remember, in those smaller populations, genes are more often lost, And the genes that are deleterious, remember those ones that are bad, okay, those numbers can start to increase. So when there are fewer copies of the alleles, you see this genetic drift impact much more so, especially in that small population. So you can see this genetic drift impact much larger in a population that has experienced what's called a bottlenecking effect, or is a founding population, has experienced the founder effect. So a population that's been bottlenecked has had, the original population was nice and large, some kind of event happened, usually some kind of environmental event, some kind of catastrophe, a very large fire, a very big disease, some kind of could be a human impact, hunting, but something has happened to dramatically reduce the population very quickly. And so when that bottleneck effect happens, you can see a significant loss of, there's no guarantee who's going to survive. For example, in the first one, there were alleles of yellow, blue, and white alleles in there. But when we took a random sample, okay, and we had those were the ones that were able to survive, okay, no yellow ones made it. So I've decreased my genetic diversity in this smaller population. This can eventually, these genetic drifts can eventually result in a completely separate population genetically. The founder effect will happen, basically, think of it almost like a colonist situation. Okay, so this example here would be lizards. These lizards were wiped out after a hurricane, and so they took on the Bahamian Islands. So they took lizards from other islands and introduced them to the Grand Bahama Island, and just basically allowed those two lizards to repopulate the entire lizard population. So I end up here with less genetic diversity here because I had a very small initial population. So I didn't have a lot of variety within the gene pool to begin with. So let's revisit these prairie chickens. Use this as an example for our bottlenecking. Remember, with our bottlenecking, we had a dramatic decrease in the population, usually due to an environmental catastrophe. So with these prairie chickens, in 1900, their population was approximately 100 million of these prairie chickens. Okay, if you look at the map here, it says 1820. See, they're covering almost the entire state of Illinois. Okay, in 1995, their population was approximately 50 birds. So in 95 years, you lost almost 100 million birds out of the population. Okay, that is a prime example of a bottlenecking effect, and that's due to human. This one was due to human impact, hunting and habitat destruction. So these birds had nowhere to live, and they were hunted, and so there is an extreme decrease in their population, and this has an impact on the offspring. Because remember, this is a random. The people that were hunting didn't care about the genetics of the birds. When they're building new houses, they don't care about the genetics of these birds. So if the birds that are left over, if any of these deleterious genes again are left in the population, their incidence is going to increase. It actually had an impact on their eggs and the ability for their eggs to hatch. So in Illinois, where we had this extreme bottlenecking event, so you had less than 50% of their eggs were able to hatch in the late 1990s, where in the early 1900s, 93% of their eggs were hatching. And when you go to the other states that did not have any sort of bottleneck effect, they still have a very high percentage of the eggs able to hatch. So, which would lead us to believe that there could have been some allele that we can basically genetic disposition for weak eggs that wouldn't hatch. And so this allele has become more prevalent in their population because their population became so small. And if a decent number of them in the small population had this allele, it's going to continue to get passed on. So looking at a founder effect example, okay, we have here the Amish community. Okay, and so the Amish community has grown in numbers. Its population now is significantly larger than it used to be. But it started from a very small group of individuals. And so there was a limited number of alleles in the gene pool. This would be an example again of that founder effect. Okay, so there's a limited number of alleles to choose from in that gene pool. And one of the, within the group, there was the allele for polydactyly, which, I can't spell, which is more extra fingers and toes, extra digits. Okay, and so with this allele for polydactyly in the original small population, okay, that allele is more prevalent in the Amish community because they haven't had a lot of gene flow into their population. Even though their population has grown, it has not grown with a lot of outside influences. So there is a higher incidence of polydactyly within the Amish community than there is in other communities because of this genetic kind of isolation. Same with those lizards that we were talking about. They're called anole lizards in the Bahamas that were used to repopulate the islands after they were initially wiped out by the hurricane. So remember, we're talking about things that are going to affect microevolution. So those founder effects, those bottleneck effects, they're going to change the allele frequency. So will non-random mating. Remember, our triangle is change. This is going to be another thing that will change our allele frequency. In non-random mating, we have a choice of the mate. And so if the gender that is choosing the mate, usually females, but if the gender that is choosing the mate is selecting for certain traits, then that's going to alter the allele frequency. Because remember, the allele frequency is going to be can change from generation to generation, but it's going to change based on reproductive success. So if the females are looking for a particular trait in the males, and those are the only ones that are able to reproduce, then they're the only ones that pass on their genes or their alleles. And so we start to see more and more, you can start to see a shift in the population towards a particular set of alleles. So this non-random mating is a form of sexual selection. So it's obviously going to impact allele frequency. And so sexual selection is just, I've got individuals with particular characteristics that are more likely to obtain a mate. So basically, again, your organisms are looking, your particular genders are looking for something in particular. So like the example over here we have is peacocks. Okay, so male peacocks have these very flashy plumage feathers that help them get chosen by the female for reproduction. It's a reproductive advantage to have this big, giant, beautiful tail for display. However, it is a disadvantage when it comes to predators because they're basically dragging that tail around as they're trying to get away from a predator. So it is it's a trade-off here, and they're increasing their chances for reproduction, but they're possibly decreasing their chances for survival. As a general rule, in organisms that do sexual selection, the female is going to blend into their surroundings better. And so the female, you can see the females in these two pictures here, the females blend into their surroundings better. So the females usually have an increased survival over the males because they're not as noticeable to the predators, which enables them to expend their energy raising the young, growing the young, or taking care of the eggs, whatever it is they need to do. They can use their energy resources in other places as opposed to constantly running and hiding from predators. Okay? So it increases the survival of the females, which again shows the importance of the female choosing the correct male. Okay, if they want to be sure that they're choosing a male that has a good balance of that will also be produce a viable, good offspring, but will also still be able to survive. And this can lead to what's called sexual dimorphism. A morph is the different, basically the different body types. If y'all are into fitness, you may have heard of like an ectomorph and an endomorph. And that's like your pear-shaped and your apple-shaped. Okay, so these are the different, basically, they look different. There's a distinctive difference between the male and the female when they reach sexual maturity. You can see that happening here in these birds. Okay, distinctive difference between the male and the female deer as they reach maturity. So there are two kinds of sexual selection. We have intrasexual selection and intersexual selection. Remember, intra is between, where inter is between, intra is within. So in intrasexual selection, there is selection within the same sex. So this is, for instance, you see all these males over here on the right here competing for a female. That would be intrasexual selection. The males are competing with other males. Okay. In intersexual selection, this is the actual mate choice. And so in this case, this is when the individual of one particular sex, usually the female, is choosy or picky. Like we saw with the peacocks, you see this with the robins here. The female lions are going to choose their male, and a lot of times that is based upon showiness. Okay? And so they have these displays, these physical displays that show that they have characteristics that would be good and that the female wants to have to be able to pass on to her offspring. So natural selection is our selection, the only kind of selection that's really going to consistently cause some kind of adaptive evolution. Somebody has a phenotype that is more advantageous to the environment at that particular time, and so over time we start to see the entire population adapt. Natural selection is a combination of chance. And so the chance is going to come from those mutations. We can't pick a particular mutation. Okay? We can't guarantee a certain mutation is going to happen, as well as what they would call sorting. Okay? And that sorting is going to be the allele favoritism. You know, there may be a particular trait. For example, we were just talking about the sexual selection. Males, lions in their large mane. Okay? The big showy mane is favored over a very small mane. And so that allele would be favored over the others. And so that would be the sorting part. So the chance is the mutation. The sorting is the actual kind of selection. What allele is best at that particular moment for that environment. Okay? And organisms then will start to exhibit what we call relative fitness. Remember, in organisms, fitness was basically its contribution to the gene pool of the next generation. Okay? That could be measured by the amount of viable offspring that it was reproducing. Okay? And its relative fitness is going to be its fitness, so its contribution to the gene pool of the next generation relative to other individuals. So basically, compared to the next guy, how did this one do? Okay? So that's going to be their relative fitness. Are they able to compete for the mate, to actually cause the reproduction, to produce the viable offspring better than the guy next door? So we have three types of natural selection. The first one we're talking about is directional selection. So directional selection is going to favor individuals of one extreme or the other. So it's going to favor, if I have a population of individuals, so this is my range here of phenotypes down here, if I'm talking math. The peak would be my mean, right, my kind of average phenotype. And so if I'm looking at directional selection here, I'm favoring, so you can see I'm starting to favor this darker, which is more on the extreme end. So I'm starting to favor one particular quote-unquote extreme phenotype. The arrow here represents selection against in the population. So something is selecting against these lighter colored mice in this particular population. It could go the opposite way. You could be selecting for these light-colored mice and not these dark-colored mice and shifting your population that way. It doesn't matter which end of the extreme, but basically you're shifting your population toward one extreme or the other. For example, if we're talking about these mice, and you see these mice have a color range of light to dark, and so maybe they live in an area that's kind of rocky, and those rocks are all pretty dark colored, and so it would favor them to be more dark colored because it would make them easier to hide from predators, from bird predators that are coming in from the air. It enables them to hide better. And so this is a directional selection. I'm favoring towards one extreme of the phenotype of the population. Okay. The impact of this is that this can lead to, okay, let's put that on a whole new slide. There we go, that's better. This could lead to, eventually could lead to a loss of diversity, right? So we could see a decrease in our diversity here, which could make the organism eventually either speciate, so become its own species, so we have speciation occur, or possibly go extinct if the environment changed. So you can see if this is occurring over a very long period of time, okay, I can see my population shift even more. My mean, you see, as I narrow more and more and more, I lose this diversity, okay, because I'm losing the phenotypes here. Okay, my frequency is increasing, and it could result in speciation or extinction. Okay, so disruptive or diversifying selection favors both extremes of the population as opposed to the intermediate. So again, remember my arrow here is for what I'm selecting against. So I don't want these intermediate phenotypes. I favor either more light or dark. I favor the extreme ends, not the middle area, with this disruptive or diversifying selection. This could be, again, if we're talking about this population of mice that live in this rocky environment, this time, instead of the rocks being either all dark or all light, maybe there's a pretty good mix of light rocks and dark rocks. And so these mice, if they're light, they can hide. If they're dark, they can hide. If they're kind of a medium color, they start to stick out a little bit more. Okay, so that would be our disruptive or diversifying selection. So our last kind of selection is our stabilizing selection. So in stabilizing selection here, we actually favor the intermediate and we remove the extremes. So we're not favoring these light or these dark. We're favoring these intermediate colors here. So you notice my mean value here has increased. Okay? My variance, though, my range has decreased significantly. So this is definitely going to decrease our variation here. Okay? And so if again, which could be an issue if the environment changed and we've lost some of that diversity there. So some of the things that populations may do to try to preserve their genetic variation so that they don't lose that variation if they're having one, especially if they're having like a stabilizing selection or a directional selection occurring, is that they're going to have some kind of built-in kind of fail-safes that will help keep that genetic variation within the population. So one of the things that we need to keep in mind is that some of the genetic variation in the population is neutral. It doesn't have an advantage or a disadvantage. So even though there is the variation in the DNA sequence, there's the differences in the DNA sequence, those differences are neither good nor bad. Okay? They are neutral. Now, if the environment changes, could that situation change? Yes, possibly. But at the current moment, they are neutral, but they still allow that variation. Another thing is diploidy. Many organisms have 2 copies of every gene, just like we have pairs of chromosomes. Okay? And what this allows for, basically it allows for genetic variation in the form of recessive alleles. So they have these recessive alleles that a lot of time are less favorable. And so this way they can remain in the population because they may be unfavorable at that time, but in the future they may be great, they may have a benefit if the environment changes. So we don't want the allele to necessarily go away. So by being diploid, they can hide, quote unquote, those recessive alleles even if they are not quite as favorable. Okay? A haploid organism, remember, haploid organisms only have 1 copy, and so haploid organisms express every single gene. So if the gene is unfavorable, then that organism doesn't get to reproduce, and so it decreases the variability, which over time, like we said, if the environment changes, maybe that trait that was not an advantage earlier becomes an advantage now, but it's no longer available because it has been removed from the population. Okay? And recessive traits are going to, these recessive alleles in the diploid organisms, remember they stay around with those heterozygous individuals, those individuals that have, one dominant, one recessive allele. Another way that the population will try to retain its genetic variation is through balancing selection. So with this balancing selection, natural selection itself keeps two forms in the population. Okay, so natural selection favors either more than two forms or two forms in the population. So it helps maintain some of that diversity in the population, and it does that naturally. We have two examples of that we're going to talk about. Heterozygote advantage as three, actually. Heterozygote advantage, frequency-dependent selection, and balanced polymorphism. So if we're talking about heterozygote advantage, we talked about this when your freshman year. Sickle cell, the sickle cell allele and malaria. So with heterozygote advantage, an organism being a heterozygote, okay, is better than an organism being homozygous. Okay, so this one is going to be better. For instance, let's look at sickle cell. So you can see, here's the cross-section here of a sickle cell red blood cell. Not great to have. Okay, so normally you would think it would be best to be this. I have no possibility of any form of sickle cells happening. Okay, but in areas where malaria is prevalent, it is actually more beneficial to be heterozygous opposed to be strictly homozygous dominant. The sickle cell allele offers protection against malaria. You can see in this map over here where you see the polka dots. There is the distribution of malaria. And you see in these areas that have malaria, we see a higher incidence of the sickle cell allele. And it's because, again, it offers that heterozygote advantage. So it keeps the sickle cell allele in the population. Okay, and so it maintains the diversity, but it does not, and it does this compromise, this balancing selection. Okay, they may have to deal with having sickle cell symptoms on occasion, but they have this natural built-in resistance to the malaria. Another form of our balancing selection, keeping these two or more forms in the population, is our frequency-dependent selection. Okay, and you're going to see me use FDS for this from now on. Okay, so the frequency-dependent selection, this is the fitness of a phenotype, and this fitness of this phenotype is going to depend upon how common it is in the population. And you can have either positive frequency-dependent selection or negative frequency-dependent selection. So with a positive frequency-dependent selection, the fitness of the phenotype increases as it becomes more common. Okay, so the fitness increases as the phenotype basically increases or becomes more common in the population. Where a negative frequency-dependent selection would be where the fitness of the phenotype is going to increase as the phenotype becomes less common. So we see less and less of this. An example with this Batesian mimicry. Remember that with our butterflies? We had the non-poisonous butterfly, the viceroy butterfly, mimicking the poisonous monarch butterfly. If those mimics are rare, birds will avoid them because when they encountered a butterfly, it was more likely to be poisonous because the mimic is rare. The phenotype is less common. So that actually increases the fitness of these butterflies because if the phenotype is very common, then the birds have had a better chance of encountering a non-poisonous butterfly. Okay, and so their butterfly's fitness would go down. So the Batesian mimicry in these butterflies is an example of a negative frequency-dependent selection. The frequency of the fitness of those viceroy butterflies increases if there are fewer of them around. So the birds are a little bit more leery of actually eating one of them. So our third kind of balancing selection, again, a way to maintain this genetic variation is through balanced polymorphism. And so with balanced polymorphism, the population itself has two distinct morphs. Remember, a morph is a type, like blonde hair or brown hair. Okay, so it has two distinct morphs in the population. And even with these two distinct morphs in the population, the allele frequency does not change. And so this can be a result of a couple of things. Okay, there could be variation in the environment. Sorry, I was making sure I spelled variation right. There could be variation within the environment where one morph is favored, and the environment may change. And so at one point one is favored, now another one is favored. It could also be a result of a time that one morph is better suited for one particular time of the year. For example, over here on the graph, let's blow this graph up a little bit for you. Okay, so over here on this graph, you can see that there are two morphs of this ladybird beetle, the red one and the black one, and you can see that during different times of the year, one morph is favored in the other. In the springtime, the red one is more abundant. In the fall, the black one is more abundant. In the wintertime, the red one, the black one is more abundant more in the summer. Okay, but that's an example of a balanced polymorphism. So it's to the organism's benefit to keep both of these morphs in the population, because at different times of the year, one morph is better than the other. The last thing that we're going to talk about here is why don't we then have perfect organisms? If evolution is happening here and where the best suited for the environment is the one that's reproducing, how come they're not perfect yet? Okay, so there's a couple things that we need to keep in mind. Selection can only act on the existing variations. So if that mutation didn't occur, then the selection can't happen for it. You know, they're limited by what mutations have occurred, so what variations are in the phenotypes. So that limits the organism's ability to become quote-unquote perfect. Evolution is also limited by the historical constraints of the organism. The species is bound by its ancestral constraints. Okay? It may have changed over time, but it hasn't necessarily dropped every single ancestral trait that it had before. And so it can only work with basically what it was given. Okay? It can't all of a sudden go from 4 legs to no legs and wings. Okay? It is bound by its ancestral constraints. Adaptations are also very often a compromise. For example, a loud mating call by a bird, okay, by a male bird, is going to attract females, but it also attracts predators. So it's a compromise. It's a trade-off. Okay? He's got this loud mating call so to help make him more reproductively successful, but he also now runs the risk of being more likely to be eaten. Also, we need to have the quote-unquote perfect storm. Chance, natural selection, and the environment are all interacting for evolution to happen. There has to have been the mutation. There has to be a selection pressure that makes that mutation better. Okay? And that's going to be due to the environment. And the environment's not static. It is constantly changing. And we can't control it. You know, there may be a big giant hurricane that comes through and blows a bunch of birds hundreds of miles away to an island, and it did not necessarily pick the birds that were best suited for that new environment. Okay, so evolution has its limitations, and we're not going to end up with perfect organisms. So we're going to be working with this vocabulary now, and then we're going to start applying this to that Hardy-Weinberg equation and being able to calculate if evolution is happening within a population.